Hello, everyone, and welcome to the State of Cultivated Meat in Australia conference. First, I'll begin by acknowledging that this event is being hosted on the lands of the Wurundjeri people, who have been the custodians of this land for thousands of years, and acknowledge and pay our respects to their elders, past and present. I also acknowledge the traditional custodians of the various lands on which you are all attending, and the Aboriginal and Torres Strait Islander people participating in this conference today. My name is Marian, the co-founder and president of the Alternative Protein Initiative, and I will be your MC today. We've also got Mars, Tech, and Nidhi from the research team helping out behind the scenes as well. So to start things off, this conference is organized by the University of Melbourne Alternative Protein Initiative. This is a student group that aims to generate interest and excitement in the field of alternative proteins at our university and beyond. We envision building a healthy, sustainable, and just food system by creating a vibrant and robust community for alternative protein research, education, and innovation, starting from the University of Melbourne. And to achieve this, we currently run various events and projects ranging from discussion groups and science programs, food outreach, social outings, and educational events. And we also wanted to mention the survey we'll be relaunching very soon, and maybe Sam and I will be collabing for this as well, is this student interest survey, which is designed to collect the student interest in learning more about alternative proteins in our curriculums. This information will help support a more long-term project in which we talk to educators about the student demand for these topics and thus advocate for the inclusion of alternative protein topics in relevant curriculums. And if that sounds interesting to you, make sure to sign up as a member for free and follow our Facebook or Instagram, where we will post upcoming events and updates on projects. You can also scan that code over there for those quick links. We're also holding a special general meeting soon to elect new committee members. So send us a message if you'd like to be involved in the committee. All right, so I think that wraps up the Alt Protein Initiative introduction. So let's get to the meat of this conference. Yes, there's going to be puns. Here's what will happen today. There will be 3 fantastic sections covering the science, the commercialization, and the future directions of cultivated meat. After all panelists speak for each session, there will be a panel discussion, and this is where we will take questions from the audience. So feel free to start adding in those burning questions in the Q&A section and upvote any questions that you like, so we can make sure we ask our panelists. There will also be breaks in between those sessions, and at 5 p.m. there will be a very relaxed networking session where you'll be joined by attendees and maybe even panelists to mingle and chat about the conference, among other things. The Zoom link for this is on Eventbrite and on the Facebook event page. All right, so let's go to our first session. We have these 3 amazing speakers, Sam Perkins, Pegah Farokh, and Nick Beaumont, speaking about the science behind cultivated meat and its technical challenges. I will be introducing each speaker before they speak. So to start, who better introduce cellular agriculture and answer the question of what is CellAg than Sam Perkins himself, the CEO of Cellular Agriculture Australia. Yes, so Sam has over 10 years of experience across the academic, humanitarian, and private sectors. He completed a PhD in aeronautical engineering and went on to do a postdoc and lecture at the University of Tasmania, where he received a Teaching Excellence Award. His expertise in participatory approaches and innovation management marries well with his experience in strategic leadership and change management to support teams create and develop novel, strategically aligned, and impactful solutions. Sam is motivated by the role appropriate technology can play in delivering sustainable, scalable, and meaningful solutions to complex challenges. He is passionate about animal rights and the rights of disadvantaged communities around the world. And so yeah, we're excited to have you here. Take it away, Sam. Thank you.
Thanks ever so much for the introduction, Marian, and thanks heaps to the team, to you and the team, for giving me this opportunity and for putting on what looks to be a very, very exciting day. I hope you can all see my screen now, which should have an introductory slide. Unless I hear otherwise, I'll assume that that's the case.
Yep, you're all good to go.
Fantastic. Thanks a lot. So before I go on, I'd like to extend my acknowledgement to the traditional owners of the lands where I'm meeting from today, the Muwinina people of Nipalona down here in Tasmania, and extend my respects to the elders past, present, and emerging. What an exciting opportunity we have. And this space, the space of cellular agriculture, the role I have at Cellular Agriculture Australia, and the ability to engage with all of you is genuinely a privilege and a huge opportunity. And so today I'm going to provide you with an introductory overview of why cellular agriculture is important, what it consists of, a little very quick flash over of the technologies that are involved, before looking into a highlight of some of the challenges facing the sector and how the sector is evolving in Australia, before highlighting a little bit of the work that CellAg Australia is trying to do and how you might be able to get involved. So our population is set to reach 8 billion by 2030. That's an increase of 18% from today's numbers, and will lead to an increase of 2 billion people over a 10-year period. Now that's a fairly staggering statistic on its own, particularly if you look at the trajectory of global population growth over the last 100 years or so. What's more startling is when you combine it with the average per capita meat consumption for people and how that's evolved over time. We've seen a steady increase in per capita meat consumption across almost all product categories, although it must be noted that there have been some isolated cases over time where there's been both a global and localized reductions in per capita meat consumption. And at the moment, we're in one of those spots in Australia where we're seeing a localized decrease in the per capita meat consumption for the products surrounding beef. But when you combine an increase in global population and an increase in the demand for animal-based protein products, what we see is a significant increase in the overall demand for animal products. And what's that led to? Well, that's led to more than 70 billion land animals and trillions of sea animals being killed and produced and processed each year to feed a population of just 8 billion people. Now, on the one hand, that's a terrifying fact. On the other hand, it's quite remarkable how our agricultural systems have evolved over the last 100 years or so to be able to keep up with this increase in demand. And it's done so through two, I guess, fundamental mechanisms. One is the introduction of technology and automation, where we've moved from the place of the farmer and the farmhand milking cows individually and distributing that product very locally to a heavily industrialized and the intensification of our agricultural systems, where those very same cows can now be milked en masse, almost exclusive of any human interaction through autonomous robots and milking systems. And that milk is then placed into a very, very large global supply chain. And so with that, we've seen this incredible industrialization of our agricultural systems and an intensification of the same system. And that has been very effective. It's been incredibly effective in keeping up with the demand that we've had for animal-based protein products, but it hasn't been efficient enough. And there are some fundamental barriers that have, I guess, thwarted its increase in efficiency. One of them lies in biology, where we have a fixed, I guess, conversion ratio from the amount of energy it takes to put into an animal, such as a chicken, and the amount of food, energy, or calories that you can get out and consume as a human. So we have a ratio of 8:1, or a 13% conversion ratio for poultry, and only a 3% conversion ratio for beef. And that sets a precedent for how efficient this system can possibly get. And when you have the industrialization combined with some of these underlying principles or factors, and this huge growth in demand that we've managed to keep up with, and we haven't just managed to keep up with it, but we've managed to do so whilst decreasing the purchasing price of many products that would have previously been considered very much a nice to have and are now a very regular occurrence on plates around the world on a daily basis. And those two things have come together to basically have the animal agricultural system contribute significantly to 4 major global challenges. And these aren't the only ones, but they're significant enough to be noteworthy. One of them is climate change and greenhouse gas emissions. The other one is deforestation and biodiversity loss. Food insecurity is a significant challenge that I think we're probably all facing and seeing on with relation to what's on supermarket shelves right now. And then public health and well-being. Now, for me, these are the drivers for why I got involved with cellular agriculture personally. And I'd like to dig into a few of these in a little bit more detail. After starting with CellAg Australia, one of the statistics that really jumped out to me, probably above all else, was the amount of the Earth's livable land surface that is taken up or consumed to support the animal agricultural industry. And Our World in Data highlights that this is 38% of the Earth's livable land surface is required to support animal agricultural practices. Now, that doesn't include anything relating to the sea or the fisheries industry. And if you look at, in contrast, the contrast between 38% of the world's or the Earth's livable land surface is required to support animal agriculture compared to just 12% for plant-based foods. That is just a staggering statistic. And that 38% equates to the equivalent landmass of North and South America combined. And you can obviously extrapolate how that is a significant contributor to a number of the challenges that we've listed above, but in particular, biodiversity loss and deforestation. Some additional facts and noteworthy points is 14.5% of all greenhouse gases can be attributed to animal agriculture. Outside of the land use, one of the statistics that really sat me back in my seat was, and depending on where you read, either more than 50% of the global supply of antibiotics goes into animal feed, or in the United States, greater than 80% of antibiotics within the United States are fed to animals. Now, that's a mind-blowing statistic because I think in the back of my head, I had, well, antibiotics are used primarily by humans. At least that's the way I've considered them to be used, but that's very far from the case. Now, that has an impact based on significant amounts of antibiotics end up in the products that we then consume. And that can be attributed to things like antimicrobial resistance and the ineffectiveness of antibiotics based on the resistance of emerging bacteria. Our current food system is inherently vulnerable, and we've seen it being vulnerable to conflict, to flood, to drought, and to temperature increases. The last of those three are inherently interrelated, as we know, and linked to probably one of the biggest challenges that we have as a global community around climate change. And something that's been a fairly hot topic globally recently is the prevalence and the increase in prevalence of zoonotic diseases and the attribution that can be made between the industrialization and the intensification of our animal agricultural processes and the increased risk and prevalence of zoonotic diseases like coronavirus. But just imagine, just for a second, imagine if we could reduce emissions produced through our agricultural system by up to 96%, reduce land use by up to 95%, reduce water usage by up to 96%, remove unnecessary suffering from both land-based and sea-based creatures, improve health and well-being and improve food security around the globe, and increase the accessibility of food to people who need it most. But whilst doing all of that, give people what they desire. Like, I've been on a plant-based diet for more than a decade. I made a personal choice to change my consumption patterns because of the impacts that I was observing from my prior choices. But it's inappropriate for me to expect everybody to make that same choice. And through the work I've done in product development, starting off with desirability around the product or the solution you're trying to produce is an absolutely essential starting point. And cellular agriculture gives us that potential. But how? Well, the opportunity is here by creating agricultural products from cells through cellular agriculture and removing the need to consume products directly from animals themselves. Now, cellular agriculture as a topic, as a sector, has been really interesting for me to enter into, and even the terminology is evolving. And so recently we went back to first principles. So cellular, from the Oxford Dictionary, is relating to or consisting of cells. Agriculture, in short, is the science or practice of farming to produce products from animals or other products related to the agricultural practices. So what happens when you put those two things together? we end up with cellular agriculture, which for us is the science of using cells to grow food and other products. Now, something that's important and noteworthy here, although many examples in this presentation are linked to animal-based products, and a lot of the dialogue that I've given you so far is based on the animal agricultural system, that isn't the extent of where cellular agriculture can play a role for us. And it has a huge amount of impact potential across the agricultural food scope or system. And by the way, the images that you will see throughout this presentation are all linked to products or ingredients that are derived from the technologies that sit within cellular agriculture. And so what are they? Well, for us, the core technologies that sit within cellular agriculture are cell cultivation and precision fermentation at this point in time. Now, some of you will have heard of cell cultivation referenced quite explicitly within cellular agriculture, and maybe some of you wouldn't. What I noticed when I joined the field was there was two categories talked about. One was cultivated meat or cultivated seafood, and the other one was precision fermentation. And so we had a product category being talked about next to a technology or a process. And so what we've tried to do is create an umbrella, which is cellular agriculture, which includes two technology types or processes in cell cultivation and precision fermentation, which are then used to produce or contribute to a number of different product categories and product types. And so at a high level, well, how do these work? This is not my background explicitly, and so I'm going to provide a very high-level flyover of these two different technology types, because you're very, very fortunate to have some incredible speakers in the lineup today whose backgrounds are very much grounded, particularly in the cell cultivation side, which is the focus of this conference. So cell cultivation, or the production of products like cultivated meat, starts with a biopsy, arguably painlessly taken from a cow in this instance, where cells are isolated from that biopsy, and the different types of cells can be isolated depending on what you want to produce. Those cells are encouraged within a media to grow and multiply until you have a sufficient number of a particular cell type, or multiple different cell types, where those cells are then triggered to differentiate or grow and turn into a particular type of tissue. Now, that could be fat or muscle or connecting tissue, for example. And in fact, maybe ideally, you want a number of different cell types to grow together around a three-dimensional structure or a scaffold, so that they grow and produce what we or some of us desire in, for example, the beautifully marbled Wagyu steak. Now, precision fermentation, on the other hand, looks at the use of microbes and the genetic engineering or reprogramming of those microbes to be able to produce a particular molecule. Now, once that genetic modification has been done, or genetic engineering has been done within the microbe, that microbe, for example, a yeast microbe, is then fermented within a bioreactor, and the yeast is programmed effectively to produce a particular molecule type, like one of the casein proteins that's required for the production of milk and other animal-free dairy products. So in precision fermentation, well, what can that be used to produce? The starting point here, which was actually, I believe, one of the really critical proof of concepts of precision fermentation, was insulin. And precision fermentation has been used to produce insulin, for example, in support of people with diabetes for decades. More recently, the evolutions in the technology and the opportunities or the necessity for the likes of cellular agriculture has seen that expand to be used to produce animal-free dairy products such as milk, ice cream, and cheese. Now, there are a number of other products that can be produced, but there's some good examples at this stage. And we've actually got a number of companies in Australia who are in the process of producing or developing these products that we hope to be seeing in the market in the not too distant future. From a cell cultivation perspective, we have cultivated meat products, cultivated fish products, leather, and more interestingly, very recently, I saw a Veg Economist article that highlighted that cell cultivation was going to be used to produce coffee because of the inherent complexity of coffee as a product. Now, I've included the concept of a liger there. And one thing that fascinates me about the cultivated cell cultivation side, or the ability to produce cultivated products, is the fact that we're not restricted to the current categories of products that we see on shop shelves right now. we're not only able to blend and bring together different tissue samples in different proportions to create different nutritional profiles, but we're also able to bring together different animal types to create food categories that or categories of meat, for example, that we just simply haven't seen before. And that, from a culinary perspective, and someone who is very fascinated by food, has just endless potential to kind of redefine not only the food system, but the way in which we perceive foods, and in particular, potentially in this category. So that all sounded like a beautifully rosy picture painted off the back of a bit of a dark and dismal starting point. But it's not quite that simple. Now, although there has been a huge amount of progress and the sector is rapidly evolving, there are a number of challenges that really face, that are in the way of seeing the sector as a whole reach its impact potential. These could be summarized, and I'm really looking forward to seeing how some of these challenges are tackled through the conference today, particularly with relation to the cell cultivation side of cellular agriculture. But there are some fundamental questions that remain unanswered and that could be a deal breaker with relation to how the sector can reach not only a point where its products are comparable on a taste perspective and from a desirability perspective, but are also able to reach parity from a cost standpoint. There are challenges around talent. Even the startup sector in Australia, which is probably really, really good news for many of you who are going through your student journeys or are looking to potentially progress a career in cellular agriculture, but there is a talent shortfall, and that talent shortfall is only expected to get worse as the sector looks to move towards scale and to commercialize its products. Now, worse is a challenge for the sector to work out, but it's a fantastic opportunity for those looking to get involved in the sector in the short, medium, and long time frames. And the capability set that's going to be required is going to change significantly over that time. There's an insufficient amount of open access research at the moment. And as more and more research is being done within companies or through bilateral relationships between a particular company and a particular academic institution, where there are strict intellectual property, agreements between those institutions, the barrier to engage in the sector and start gets increasingly harder the more time passes. And that has flow-on effects to challenges around the future competitiveness of the industry if one or more particular company gets a sufficiently large head start. There are significant challenges right now around access to infrastructure and manufacturing capability, particularly as we look at moving towards from sort of bench scale production, where we've got proof of concepts and prototypes being developed really, really well now, through to the scale that can support commercial production and really drive down costs and start to move towards having a significant impact on our food systems. There's generally a lack of public awareness, and the Alternative Protein Initiative at the University of Melbourne is a fantastic example of a solution around this, among other things. We need to build people's awareness, both from a student perspective as well as across the general public. And that's going to be critical for making sure that the public perceives cellular agriculture in a positive light. It's the same with government, but maybe with a slightly different set of messages. Government need to be aware of the potential, maybe from an economic standpoint or from an impact standpoint, with relation to how cellular agriculture could really accelerate Australia's impact on things like the Sustainable Development Goals. And lastly, in fact, not lastly, but lastly on this list, is we have a largely untested regulatory environment around novel foods that can be produced through cellular agriculture. I will highlight that there is one example that has gone through FSANZ, which is the soy leg hemoglobin produced by Impossible Foods for their Impossible Burgers and other products. And that was that has successfully passed through the FSANZ process, which is fantastic because it sets a precedent with relation to how novel foods or ingredients produced through precision fermentation have gone through that process, which has now been documented and made publicly accessible. But it is largely untested for other products produced from precision fermentation, and certainly it's untested from a cell cultivation perspective, although we believe that there will be products produced from Australian manufacturers heading down that pathway in the very not too distant future, which is really exciting for the entire of the industry. So what's happening in Australia? I'll give you a really quick flyover here because I'm conscious of time. The sector in Australia is expanding really, really rapidly. In fact, as an organisation, it's something that we are really trying to keep our finger on the pulse on. And it's actually difficult to do because of how much is happening, and not all of it's visible to everybody. And so we've got this industry map that we've put together that we've evolved for a white paper that we're going to be launching next week. So I highly recommend you have a look at that in more detail. And that really presents the economic case for the sector and the potential that it could have in Australia moving forward. So what do we do at CAA? Well, we have at the moment a number of core focus areas. One is around outreach and education. So building awareness around cellular agriculture and helping build fundamental knowledge and understanding of it and the core technologies that sit within it, as well as the impact that it could have and how people could move towards having a career in the sector, both now and into the future. We look to advocate on behalf of the sector around core messages that members of the sector want to see carried forward. And our white paper is a really good example of that, which will be launched, like I said, next week and on Wednesday, in fact. We facilitate cross-sectoral engagement, bringing together academics and industry, investors and government to talk about common challenges and identify opportunities and solutions that can be used to move the entire sector forward to benefit everybody, ideally equitably. And we have seed grants to support early access or open access and early research, and to bring more people into research around cellular agriculture, but also importantly to support academics pivot who are in proximate fields to cellular agriculture, which are going to be essential as a cross-disciplinary network of skills for the sector to move forward on. So what can you do with us if you're interested? You can learn. A couple of weeks ago, we launched a learning management system, which with Australia's first online short course. This QR code will take you to the LMS. Feel free to register, it's all free. And we'd love to hear any feedback you have on that course. You can volunteer and be part of our team. It's fantastic to see a number of our volunteers are also engaging with the Alt Protein Initiative. If you're interested in that, get in touch with me. We've got heaps of opportunities for you to explore the way you can contribute to our impact as an organisation and move towards a career in the field. Provide pro bono support or simply donate to support the future impact of our organisation. Lastly, if you're interested in this space and you want to explore it more and understand what your career could look like and the decisions that you might need to know now to make sure that you're appropriately skilled and positioned to be a valuable contributor to the sector moving forward, head to Pathways, which is an interactive tool which allows you to explore majors that are highly relevant to the future of the field, as well as STEM and non-STEM based problems that need to be solved if the cellular agriculture sector in Australia is going to reach its impact potential. And I'll leave it there. Thanks again for having me, and I really look forward to engaging with you all through the Q&A after the next couple of fantastic presenters.
Thank you very much, Sam. Thank you for the introduction. Yeah, definitely check out the CellAg Australia resources. They're fantastic if you're, ready to start your CellAg career or just, looking forward to it. I even volunteered for CellAg Australia myself, and it was a fantastic experience. So would definitely recommend that for everybody too. All right, so let's go to the next speaker, Nick Beaumont. Who will be talking about culture, medium, and growth factors for making cultivated meat. All right, let me introduce you. So Nick will now talk about this, and he is the founder of Heuros, one of the first companies in Australia creating GM-free, animal component-free growth factors to make cultivated meat. The medium they produce also does not contain antibiotics or hormones. So yeah, very excited to hear about what you do and more about Heuros. Take it away, Nick.
Thank you very much for the invitation. It's great to talk to the University of Melbourne Alternative Protein Initiative. I'm going to be talking today about the medium that we culture cells in, the growth factors that we use to stimulate their growth for making cultured meat, or indeed cultivated meat. I don't want to, I'm not quite sure about the terminology these days because our company is Heuros and we're based in Brisbane. As Sam was saying, really the technology has been known for several decades that you can take tissue samples from animals and you can dissociate them and take the stem cells out of them and grow them within the laboratory. You grow them in a nutrient-rich medium and you make sure there are no bacteria or yeast present in there. It's under aseptic conditions. This can happen for muscle cells, and you can produce milligrams, perhaps grams of tissue. But these cells only divide a limited number of times, perhaps 30 or 40 doublings. You can take some of these cells and if you're, you can try to make cell lines, which in theory at least will continue to divide indefinitely. But they've still got to be grown in this medium, and that's what we're going to be talking about today. Because the idea behind cultivated meat is that you can scale up from that laboratory practice, scale up vastly so that you can produce tons of meat and actually produce important amounts of food. So as you say, the kind of, we have to examine the details there, what's in media at the moment, and whether we'll have to change it when we scale up. So on the right-hand side, you see a bottle of medium that we use in the laboratory for growing muscle cells. It's a medium called Delbecco's Modified Eagles Medium, or DMEM. And it's sterile, so there are no bugs in it. And the reason is because it's full of nutrients or that the cells could use. So bacteria or yeast would grow very quickly in this medium if they were allowed. It contains about 30 ingredients, and I think we should actually look at the composition of the medium. And when you do, you see that, well, it's mostly water. It's about 98.5% water. So the actual solid components of the medium only make up 1.5%. If you consider muscle is about 75% water, but then that means that it's about 25% non-water. So you have to get those components from somewhere. If you extrapolate up from tissue culture, it would take about 20 litres of medium to make, to provide enough growing medium for 1 kilogram of meat to be grown. So if you extrapolate that up to producing tons of meat for the industry, then it would take 1 million litres to produce 50 tons of meat. You can see that the vast volumes involved. And really, you've got to get the price down to about 20 cents per litre. At the moment, if that bottle of medium that I just showed you, 500 mils, for about $30. You could buy the powder and make it up yourself, add the 98.5% water yourself and sterilize it, and that will cost you about $3 a litre. So there's still quite a way to go. Anyway, apart from the water, what else is in there? So if we discount the water for a moment, let's look at the solid components. We can see that about half of it is common salt, sodium chloride. And another third of it is the common sugar glucose, which provides the metabolic energy the cells need for their growth. But the actual components of growth, the amino acids, the vitamins, minerals, and metabolites that the cells need, they only make about 0.5% of the weight of the media. These media, there's several of them, and they were formulated decades ago, and they're really useful for growing cells in at small scale. But they're often supplemented with animal products, as I'm about to discuss, and so they probably need to be optimized for use if we're going to use them in the production of cultivated meat. So here are some of the kind of challenges and opportunities that media presents for the nascent cultivated meat sector. As I said, the formulation will probably change from what we're using in the laboratory at the moment. The ingredients, the source of the ingredients will actually change, because at the moment we're using reagent grade ingredients, which are very pure, but do they have to be that pure for food production? Food grade ingredients would be cheaper. Perhaps we could even use hydrolysate, which would and then supplement that with whatever nutrients need to be added further. Another point we should consider is the amount of media that's going to be used in any large-scale cultivated meat production. As I say, you need probably about 1 million litres of culture medium to produce 50 tons of meat, which sounds like a lot, but really, as Sam was saying, there's vast amounts of meat consumed. So what are you going to do with that used media? First thing you need to know is that animal cells don't use all of the nutrients that are presented to them. They, as it were, leave some food on the table, unlike, say, yeast or bacteria, which would consume the whole lot. So you don't want to discard the medium and waste that. What they do is they excrete into the medium things like lactic acid and nitrogenous waste such as ammonia. So if you're going to recycle your media, you're going to have to scrub it to remove the lactate and ammonia, and also you're probably going to have to fortify it with further nutrients so that you can use it once more in the process. The other option is perhaps to repurpose it. So for example, we could use conditioned media, the media that's been used to grow muscle cells, and use that for some fermentation or precision fermentation. So these are some of the options that still exist and have to be explored by the sector. So if that's medium, this kind of liquid containing all these nutrients, you just add it to a muscle cell line and the cell line grows, you might think, but that's not the case. Here's a case of the quail muscle cells, and they're grown on the horizontal axis for 3 days over a time course. On the vertical axis, you're looking at the number of cells, and you can see there's no increase. And this is because the growth of animal cells is regulated, unlike, say, the growth of bacteria or yeast, which just given enough nutrients will grow as quickly as possible. So what are the mechanisms of the regulation of animal cell growth? Well, there are things like cell-cell interactions. So when cells are cultured, if they're confluent, that is, if they form a continuous layer, they usually stop dividing. Our cells are also, muscle cell lines are also dependent on mechanical clues, such as, for example, our cells are adherent. They have to be in contact with a surface. They have to be stretched out, otherwise they don't grow. But probably most important that's been followed up is the biochemical cues, such as growth promoters. These have traditionally, in the years of tissue culture, they've usually come from very young, unborn animals. So in some cases, it's been cell embryo extracts, but most commonly, it's been fetal bovine serum. And fetal bovine serum is it's on the right there. It's a distressing product in that it's obtained when a pregnant cow is slaughtered. The fetus is then taken and bled out. The blood is clotted, and then leaves you with the serum. It's very good if you add it to your medium, say 10% fetal bovine serum. It's very good at growing a wide variety of cells in culture, but it is expensive. It's about $1,000 a litre. And if you wanted to make, say, a burger's worth of meat, about 150 grams worth of muscle, it would take about $300 worth of fetal bovine serum. So it's obviously a non-starter financially, but obviously you wouldn't want to base the whole sector on it anyway. So for the cultured meat sector to grow and develop, you really need to replace fetal bovine serum as the stimulant of growth, and you want a safe and affordable range of alternative growth promoters. So where would you find these growth promoters? Obviously, you can analyze your fetal bovine serum and look for those things that are present in the very early animal when it's growing at its most rapid. And there has been some interesting work showing that the most abundant protein in serum was serum albumin. It actually encourages the growth of cultured cells. But most of the work has been to look at far less abundant proteins called growth factors. Now, there's a number of families of growth factors, such as fibroblast growth factors, insulin-like growth factors, transforming growth factors. These are just some examples. They work at incredibly low concentrations. They're very potent chemicals, and they bind to specific receptors on the surface of cells, leading to a cascade of intracellular signaling, often leading to cell proliferation, the growth of the division of the cells and their growth, and a number of other activities. In some ways, they seem ideal for the cultivated meat sector. You just have to find them. You could either purify them from a biological source, but as we saw with fetal bovine serum, that's not really a starter. But what a number of companies, there's probably about 20 companies around the world, and 90% of them are doing, is they're using genetic engineering to produce growth factors. This is a diagram depicting that. So the DNA encoding a growth factor is here depicted on the left-hand side in green. And you can take that and splice it into a loop of DNA, a vector or plasmid in red here. And then you can insert that into the cell of choice, say bacteria, yeast, plant or animal cells. And these are then genetically modified organisms, as you see in the center. They can then be grown, and once you have enough cell mass, they can be induced to express this growth factor protein, as we term it a recombinant protein. The idea is then that you purify your recombinant genetically engineered growth factor away from the GMOs, and there should be no contamination. But of course, there's a concern there. There could be contaminating DNA. So let's consider whether that is kind of acceptable. Well, as an example, let's use fibroblast growth factor 2, FGF2, which a number of people are looking at using to create cultivated meat. It's incredibly potent, so just a couple of grams of it in 20,000 litres, 20 tonnes of media could be used to grow a tonne of muscle. Incredible potency, but it stimulates not only muscle, but non-muscle cells to grow and divide, including, for example, the epithelial cells that line the gut, the gastrointestinal tract. FGF2 signaling has also been implicated in the progression of cancers of the GI tract. So there has to be concern about whether this should be used in food production. The kind of doomsday nightmare scenario is that the DNA encoding this your growth factor could remain in the cultivated meat product at the end. Would some of the DNA survive the manufacturing, cooking and digestion? Well, most of it wouldn't, but DNA can survive the high temperatures of cooking, and some individuals, their digestion is less than complete. For example, the very young, old and sick, so those who've undergone a gastric bypass, for example. If DNA encoding a growth factor did get through the stomach, then it might come into contact with the bacteria in the gut, the microbiome, and a lot of these are known for taking up foreign DNA, and they may express it. So the kind of worst case scenario is that DNA encoding a mammalian growth factor might be expressed by the bugs in the gut, and this might lead to some growth in the GI tract. Perhaps the first two lines of this syllogism, some might think are extremely unlikely, but I don't think they're impossible. And when you consider that the exposure to the food source, the cultivated meat, there could be millions of consumers, hundreds of millions of consumers as the sector matures. They could be eating it on a regular basis, perhaps daily, for a long time, years or decades. And so the actual exposure to this could be significant. So it is a possible concern, and so at Heuros, what we're doing on the kind of precautionary principle is we're not using the genetic engineering approach to make growth factors. We're making our growth factors chemically. No DNA is present, so we don't have to rely on removing it later on. And we believe this is, well, it's an alternative approach. We believe it will appeal to consumers and regulators more. Here's some work from our laboratories. This is looking down the microscope at some muscle cells, a mammalian muscle cell line. On the left-hand side, you can see that we seeded these cells at 20% confluency. And after 4 days, they remain at 20% confluency because there's no growth in just medium alone. They're not stimulated to grow. Whereas on the right-hand side, the same cells were seeded at the same density, but there has been growth when they've been exposed to the Heuros growth factor. We can actually quantify this. So these were mammalian cells. This is that bird cell line that we were talking about, the quail muscle cell line. As we said before, the lower line, when the cells are just grown in media alone for 3 days, there's no apparent growth. Whereas the red line is showing what happens when the same cells are grown in the same media but exposed to Heuros growth factor. You can see there's a significant level of growth there. So to toot our own trumpet here, we have made progress. We're now growing cells serum-free, as are others, but our approach is to use it is we're not using genetic engineering at any stage in our work. Our technology grows mammalian and bird muscle cells, which will be a commercially important species. But our technology doesn't stimulate the growth of epithelial cells. We've created a minimal chemically defined medium that only contains 4 components. And I think that's the simplest medium for growing any animal cells. The next simplest that I know of is Essential 8, which has 8 components, including 4 recombinant proteins. So to summarize our talk then, there is already available medium for growing cells in tissue culture, but normally they've been used with fetal bovine serum or other animal extracts. So there are opportunities to change the formulation, to source the ingredients, and to recycle and reuse the medium, which is going to have to be done because they can't all be discarded. When it comes to supplementation, I think we all agree that the FBS has to be replaced. And I've raised some issues about the growth factor strategy, whether you should use genetic engineering to make those particular proteins. And thank you very much for giving me the chance to talk about our work here at Heuros. Thank you.
Very much, Nick. Thank you. Oh yeah, by the way, cultured meat, cultivated meat, cell-based meat. Yes, exactly the same thing. We have different ways of saying it, but yeah, totally cool. And thank you so much for sharing your approach with us, what Heuros does. And it's super cool that it's without genetically modification. And I guess it's very important as we look forward to scaling up cultivated meat production and yeah, to make that, the culture, the medium, so that it can grow a lot of cells and that it's also safe for consumers as well. So thank you very much.
Thank you.
Thank you. All right, let's move on to Pegah, Farokh.
Yes.
Hopefully I'm saying that right. Is that all right?
Yes, exactly. Thank you very much, Nick. So let's
Wait,
start.
can I introduce you?
Yeah.
Wait, I will introduce you first. Hello, Pegah.
Hello. Yeah.
We'll talk about texture and taste. So
Exactly.
yes, Pegah is a synthetic biology scientist at Nourish Ingredients. Pegah originally trained in plant cellular and developmental biology as a PhD candidate, and then as an active researcher, she worked for the ACT Department of Crop Adaptation. She then joined CSIRO in a photosynthesis group studying increased carbon dioxide concentrations in genetically modified wheat and tobacco in collaboration with ANU. Pegah's strong interest in deep metabolic molecular concepts also led her to join the plant oil engineering group at CSIRO, where she was an active member of a team producing novel food ingredients. Pegah is now a key member of the food lab at Nourish Ingredients, a deep tech company using science and precision fermentation to create animal-free fats for the next generation of delicious plant-based foods that looks, cooks, and tastes like the real thing. So thank you very much, Pegah, and take it away.
Thank you very much, Nick, for all the information. So let me share the screen. Yeah.
Perfect.
And
yeah.
do you have Nick? Okay, so maybe we can mimic this one first. Okay, thank you very much. Thanks. Okay, so we can start. So today we want to talk about the texture and taste in meat alternative products and the impact of Nourish fats. Yeah, so my name is Pegah and I am working in Nourish Ingredients company. So who is Nourish? Nourish is a deep technology company, and we are a passionate group of scientists using science and precision fermentation to create animal-free fats and eating experience for the next generation of tastier alternative proteins. As you might know, fat is the key component of food, and it determines the overall eating experience of the product, such as taste, mouthfeel, and cooking. So therefore, in some way, we can consider fat is the future of food. So what is our purpose in Nourish? Our purpose in Nourish is ignite the future of food with our novel fats by transforming the experience and making food more sustainable. From that purpose, we have 4 key missions. So these include taste, sustainability, world-leading science, and innovation. Whenever I talk about the taste, we want to elevate the eating experience of the next generation of food. When talking about the sustainability, I mean, our mission is to offer a better for the planet source of fats for the products, creating a more sustainable food system. And when we want to be the world leading science in synthetic biology, precision fermentation, and food development. And actually, and lastly, but not least, the importance, we aim to be the discoverers and explorers in ingredient innovation. So alternative meat products. Alternative meat products are an expanding market which grows sustainability during the last few years. And alternative meat products include cultured meat and plant-based meats. And meat alternatives may have the potential to address substantial global problems. As we know, they can improve animal welfare or increase food security, and definitely they can help us to decrease environmental impact and mitigation of climate change. So actually we need to have the culture meat and a plant-based meat because of that important reasons, but that definitely is challenging. So whenever we want to talk about the challenge of the culture meat, definitely as the first is cost. So it's very expensive. And the second one, the challenges is to method for the scaling up. And the third one, which is very important as well, it's taste issue. So because of the culture meats, they have lack of fat and bone, so they don't have the same taste and same texture. And actually, it can have effect of the customer interest. How about the plant-based meat? What is the challenges in plant-based meat? Plant-based meat products, as we know, they are a growing market these days. But despite these billions of dollars of investment on this area in plant-based meats, unfortunately, again, we have a taste issue in this area. But whenever we want to compare the culture meats and plant-based meats with the real meats, so we can see, for example, if you want to take a look on the steak, so animal meat doesn't create an unmistakable sensory experience, right? Because fats play a critical role in animal meats. So for this one, as a piece of a steak, even the uncooked one is a very delicate, especially with those white marbling particles between tissues. It looks very pretty seductive. And during the cooking, whenever you start the cooking this steak, you can sense the amazing smell, which can hit your nose. So, but as we are working in the food lab, every day we are working with the plant-based meats. So you cannot have, unfortunately, this experience and have a replicate that experience in the plant-based meat. And actually, in Nourish, we have made it our mission to do just that. So our mission in Nourish is to create the taste and texture exactly similar like the real animal meats. But how? So in Nourish, we use science and precision fermentation, and the process that is very similar to brewing beer to create fats. That means plant-based food can look, cook, and taste just like the real things, real meats. So by engineering the yeast genes and combined with the fermentation process, we can precisely mimic the molecular structure of animal fats, but without the use of fat, actually. So after we design the fats for ourselves in the molecular lab, then in the food lab, which these days I am working in the food lab, we are using the Maillard reaction on top of that biomass and fat. And Maillard reaction is a series of chemical reactions that are responsible for the wonderful color, taste, and aroma changes we experience exactly during the cooking process. And when you add the Nourish fats into that process, when you're cooking the plant-based foods, our fats can completely transform the experience. So by Nourish fats, you can create exactly similar like real meat taste and aroma. But the process, as I mentioned, beginning to end is animal free and is what makes actually Nourish unique. So in food lab, it's a very, maybe it's very simple sentences. Okay, so maybe we have biomass which contains the fat and we are using the Maillard reaction on top of that. But actually, whenever you're in the lab, practically it's not like this. So we need to, not only in the molecular lab, it's very challenging and very hard. We need to work on that. Also in the food lab, every day you're working very hard because actually we need to have an establishment of the Maillard base. We need to work on that. We need to have the optimization for the Maillard base for our NI materials. And also, as I mentioned, we are screening and selection of the suitable fat containing our material, which we are looking for, I mean. We are actually doing the fractionation and recombination for reverse engineering. And whenever we found, okay, so this can be have a good result for us, then we can apply the product in typical plant-based products, as I mentioned, and culture meat to see everything is work well or maybe not. So if you want to a little bit talk about in details in screening and selection of the suitable fat, and number 5, application in typical plant-based products. In this slide, we can explain more. So about the screening, we are working on actually on several biomass, sometimes yeast, sometimes fungi and bacteria strains. It depends. And also our investigation is continue, and in comparison into the Maillard base. And after that, whenever we found everything is okay, we have the sensory test. after we added the Maillard reaction on top of it, we have the sensory test. We have the sniff and taste test with the expert people in Nourish. And after the sniff test, we can apply it that complex mixture on top of the TVP flakes. If we found the TVP flakes, for example, which one has the best taste, best sniff, which one is very close, you know, maybe sometimes it's very meaty, sometimes it's very chickeny, so we have, or sometimes fishy, you know, it depends the fats we are adding on top of it. And whenever we found we like that, we do the chemical composition analysis on them to see how is the fatty acid profile, or how is the amino acid profiles, or volatile analysis by using GCMS actually, to see what is the changes in fats. So in our lab, we have another section, which is prototype section. So in prototype section in food lab, we are applying our product in plant-based product, and we are trying to have a prototype. As we can see, we are trying to make the meatballs or burgers. It depends, or sometimes we are applying on TVP. For the first time, definitely we applied on TVP, and when we found the result is okay, then we can add it on the different kinds of plant-based meats or culture meats. This is our tasting sessions in Nourish, and as we can see, we have actually science and fun in Nourish, and sensory tasting in Nourish, and very, very interesting experience. So thank you very much for your attention. Please, if you have any question, please let me know.
Yep, that's right. Make sure you put your questions down in the Q&A section, as Pegah says. Thank you so much, Pegah. It's
no
really
worries.
Oh, yeah. I mean, fats determine, you know, the sensory experience of consuming cultivated meat. So it's super important, I guess, also for the acceptance of these kind of, you know, alternative meat products. So very important work that you're doing. Also, the taste testing sessions sound really fun. Hopefully, we're
exactly. Yes, yes.
Get that. That sounds really awesome.
Yes.
All right, so I will now move on to the panel discussions. So make sure you submit your questions if you haven't done so already. But I guess I can kickstart it first with a question. And you know, when Sam mentioned there are many components that come together for a cultivated meat product. So this is a question for Pegah and Nick. Well, Sam as well, if you want to chip in. Do you think your products will interact with each other moving forwards? Do you see collaborations within different companies in Australia or beyond to make a successful cultivated meat product?
Can I explain about that?
Yeah. So because yeah.
So actually, in Nourish, as I know, we are working with V2, and we tend to have a collaboration with Impossible company and 3 or 4 another companies, but these days we have the negotiation. But for V2, definitely we have the collaboration with them. And they were very interested about the Nourish products. But so yeah, we have the collaboration. But sometimes they want some changes, or sometimes we want some changes about our products, but so they are coming here. So sometimes we are going there to, you know, think, totally think, you know, together very in a narrow road. But yeah, we have the collaboration with them. Yeah.
Yes, just following on from that, I was going to say ours is a business-to-business model. That is, there's no point us holding, for example, any intellectual property if we're not using it. So really, yeah, we have to kind of like negotiate and get on with other companies in the sector, and it's great. That's what we're here for.
Yeah, I've found that really interesting, the kind of difference between the B2C and B2B, so business to business versus business to customer. And I think there's a really interesting space to watch, which is early on, particularly when profit margins are potentially unknown as companies look to commercialize and scale products. The idea of trying to control a large portion of the value chain, from arguably creating media formulations through to packaging up final goods and distributing them to consumers. And as that becomes more known, and potentially as scale allows for greater profit margins, both from a supply chain perspective, but also as a producer, there will arguably be more opportunity and room for those business to business engagements. Whereas at the moment, I feel like there's a little bit of kind of hedging around the more you control of that value chain, potentially the more of the profit margin you seek to benefit from. But there are some obviously some challenges there, because the amount of technical depth that both Nourish and Heuros can go into can't be compared to what can happen within a small startup cultivated meat company, for example. So there's some really interesting playoffs that need to be considered. But it's just something that's really stepped out to me, looking at actually how much of the value chain these cultivated meat companies and other companies are trying to control at this stage.
Yeah, and I think, as you know, Cellular Agriculture Australia as a hub for all these different, you know, stakeholders, companies. So I think it's really important, you know, to join all those different companies together and go ahead so we can collaborate and make those cultivated meat products successful. So thank you for that. We have a question in the audience. It says, what kind of role do you think academia will play in conjunction with industry in the future of this field? I'll pass it on to Nick.
To
Nick.
Oh, right. Yeah, absolutely. A lot of the knowledge that we're working with was first created in research institutes and university research labs. In Australia, in all countries, if you're going to search for academic funding for this, you're really up against biomedical research. And so you've got to make a very winning case to your funding body for that, and to, you know, effectively to the voters and the public. But yeah, we're going to be working with universities where possible. I would say, just on a minor note, if you're in academia and you're looking to spin out a company, you know, maybe the first port of call should be the people at Main Sequence. They're really set up in this country. You know, Phil Morley, look him up online, and you can't go wrong with that.
Fantastic. Pegah?
Yeah, it's very important, Marianne. It's very important. For example, in Nourish, you know, everybody. I don't want to say everybody. We are in one field. We have chemists, plant biologists, different kinds of or food scientists. Different. It depends. You are working in which lab, but I want to say, in overall. It is not depends what is your field, you know. Sometimes for example, in Nourish, sometimes you know about the molecular, you sometimes in one person I am talking, sometimes you need to know about the engineering, metabolic, and food, and about the, you know, fats, And so it's very important. I think it's very important these days to be, to be very educated about the, sometimes not to in one area. It's better to have from a different area. It's the best, I think. So it's very important, Marian. Yeah.
Okay, thank you. And Sam, if you wanted to add something as well.
Yeah, I think maybe a slightly different bent on that is that I think the role of academic institutions really needs to be in obviously the creation of new knowledge, but making that knowledge accessible. And I think that's something that we're seeing, that like the fundamental role of academia is starting to shift away from, to a certain extent, away from this open access knowledge base around new knowledge that's discovered, through to much more commercially oriented partnerships where intellectual property is almost inherently tied between bilateral or in some instances multilateral arrangements. So I think there's a strong importance for like public funding from the likes of the ARC to support cross-disciplinary, cross-institutional research that is then made accessible early on, to avoid the kind of potential monopolization of certain parts of this industry, among others. And so that we move forward and have a genuinely competitive sector in the future, which is ultimately going to be beneficial for, I think, all of the companies involved, but in particular for the sector as a whole and the way it will be able to drive change. I also think from a research standpoint, because of, I guess, the inherent technical nature of a lot of the challenges that need to be overcome, researching getting students from an undergraduate level into masters and on into PhD, and thereafter, that's going to be an essential part of the future talent pool for the sector, where companies are really tackling some fundamental research-based questions themselves, which is remarkable, particularly given the pace that they're able to do that at. That's really surprised me, and I think, I think personally, academia has a huge role to play in this sector in the future.
Perfect. And I think I also have another question from an attendee asking if there are any data-related technologies in this industry. Are there?
I might jump in there with just an example that I think is relatable and that really impressed me. In the, I think, Cultivated Meat and Future Food podcast by Alex Shirazi, there's an interview with Suresh Paul from Vow. And Suresh really talks about the approach that he's taken at Vow in leveraging technology, and in particular, the likes of liquid handling robots and digital microscopes, and the processing, I guess, and AI that they're using within this, I guess, what would have typically been a very manual, hands-on visual process. And the data sets that they're able to draw from that approach are not only shortening the timeframes they need to identify the best cell lines, but to be able to manage, or view the evolution of these cell lines over time and collect all that data, rather than that data just be, in the head of the scientist, where a portion of that, arguably the good bits or the very bad bits, get reported. Everything gets logged and can then be accessed in the future. So I think that's a really interesting example of where we're seeing a whole range of different technology types that typically don't sit within what can be our reference, which is sort of cell biology. Well, this is a long way away from cell biology, but it's really being applied to enable what the future of, I guess, cultivating cell lines might look like.
Yeah. Does anyone want to add more to that, or should I move on? Okay, great. I think this is a question for Nourish. Will you consider the job application for someone who's working in brewing industry and now to explore the precision fermentation technology? I guess
Yes.
talk about jobs.
Yes, of course. Yeah, Nourish, has the open arms for the younger generation, not for the fermentation. Fermentation lab, we need, help, or in the molecular section, we need help, or in the food lab we need, if you're expert in one area or two areas, yeah, please apply for that one. Yeah, so Nourish always, it has the open arm for that. Yeah,
A lot of
please
job
do.
opportunities.
Yeah, please do. Yeah.
Yeah, fantastic. All right, since we're talking about precision fermentation, well, we're talking about another topic. Do you think there would be a better consumer perception of technologies like precision fermentation as compared to what it has been for GMOs? So I guess, would that be to Pegah, maybe?
Sorry, what was your question? What was the question?
Yeah, there's 3 upvotes on this question, so people
Yeah.
really want to know. Okay.
Yeah.
Do you think that there would be a better consumer perception of technologies like precision fermentation as compared to what it has been for other GMOs or GMOs?
I think we need the GMOs. Then we need to use the fermentation for scaling up. And beside the GMOs, these days, because sorry, and we have the mutagenesis lab as well in Nourish. So definitely, we need to work on both. So we cannot say which one is more important in Nourish, or we cannot say molecular techniques is very important, fermentation is very important, or food lab is very important these days, or fats analysis. In Nourish, all of these 4 labs are very important for us, and we need to work on it. And about that, your question about the data science, I need to say it's very important, because at the end we found it's very difficult for the scientists if personally we want to work on the data. For example, I remember whenever I was working in the molecular lab, I needed to screening the colonies by myself one by one. But these days we are working on the, have the robotic help for that one to do it more smoothly and faster for screening the colonies. So faster we can find, okay, so which one they have our target gene. So yeah, so I think, yeah.
Okay, thank you for that. All right, because we're talking about GM, now let's talk about GM-free media. So this is to Nick. How else, this is a question. It's good to know that the media is GM-free. How else can you convince someone who is a critic of cultivated meat or apprehensive about this?
Sure, information. You've got to be open at some stage. I was talking with a startup in North America, and they were saying they didn't want to go through patenting their technology. They were going to keep it as a trade secret. I think that's the wrong approach. I think you're better off trying to convince people by giving them the information. When I was saying about we're not using genetic engineering in our production, I think this is specifically about the case of growth factors. They are such potent molecules that I really don't want to see their DNA getting out in the wild in microbes. That's why I'm making this point. But it has to be said, there's probably about 3,000 academic surveys of people's attitudes to GM technology. And I think it's fair to say they haven't embraced it. And it's been around a good kind of 30 years. It's just never appealed. People have been told by the highest authorities that it's safe, but it never seems to have really grabbed the public imagination. So better people than me presenting these arguments can't persuade people. Perhaps that, I don't know what the way is forward. I personally think that we don't have to use genetic engineering in the production of cultivated meat. We're going to avoid it.
Perfect. A few more oh, sorry, go on, Sam.
Yeah, just to add on to that, I think there's a really interesting, and we've had a number of conversations, including with the likes of Fasanz about this. I think there's a really interesting and a really important distinction to be made. And communication, because I think you're right, Nick, that awareness raising and education is essential. But the critical difference between using genetic engineering or genetic modifications to influence a production process that creates a protein molecule that unto itself is not genetically modified, similar to using genetically modified ingredients in a media, but the end result cultivated product is arguably not genetically modified unto itself. There's a really fine line there. And I think the communication around that, like, is the product that people are consuming genetically modified? Well, it's an interesting one. Whereas, or does it come from a process that inherently requires genetic modification? And I'm not saying it inherently requires in the cultivated cell cultivation side, but where it does, that I think is a really important piece for the sector to get across how it communicates that to the public, if indeed it decides to go down that route. Because I think that's a really critical distinction that isn't always made explicitly.
I agree. And in the precision fermentation side, we have like Perfect Day in America, who produces, dairy-free, well, animal-free dairy proteins, and they're used, made using GM, organisms, but the product itself isn't. So I think, that's a really important distinction to make with cultivated meat and other conversations with food. So, all right, so I think we have more specific questions. This is a technical and science related session, so I will go ahead and ask those. This one seems to be for Nick. Is it possible to make media for both epithelial cells and muscle cells to grow in, or do they need different factors?
The media, it contains most of the same ingredients at slightly different ratios. So there are different media, but they all contain the amino acids and the nutrients that cells need to grow. The growth factors that specific cell types have, there is quite a lot of crossover. So a lot of the growth factors that I was discussing have a very broad specificity. And that's one of the concerns we have about using them in food production. But yes, there are ways you can grow. For example, there are, we think we've identified some more or less muscle-specific growth factors that we can use that won't have an effect on epithelial cells. We've not tried to grow epithelial cells at this stage, but I mean, people who are involved in, say, skin grafts and things like that are probably more, you should, research that sort of area.
Okay, and also this might be a question for Nick and Sam. Are there any companies in Australasia working on the recovery of used media for purification for reuse as well? Oh, there are.
Oh, no, I just think it's a great area. I think this has to be, it's not an add-on that we're going to, once we get to bulk, we have to kind of plan this in at an early stage. To my knowledge, I don't know of anyone, but I think it's I think there's some interest in the CSIRO. But perhaps possibly Sam knows more about that.
I don't specifically, unfortunately, but I agree. It's a really, really interesting discussion point, and I think it comes into, what might be for the sector and the potential impacts. It comes down to a whole heap of choices that the sector is going to have to make, and this could be one of them, as well as the reuse of things like water and the way in which we source electricity.
Absolutely. Okay, there's two more questions. I want to try to fit them in. I know we're a little bit pressing on time. So this one is for Pegah. If I'm not mistaken, in her slides, it said that sugar and yeast are used and fermented to create the vegetarian fat. However, as we know, a high intake of sugar isn't so good. Is there a compromise between having the taste and texture through the meat compensation product, a meat alternative, and health, or is sugar in this fermentation process not too harmful to worth thinking about?
She or he is correct. So for the Maillard reaction, we are using of protein, sugar, and add it on top of the our fats. So that's why we need to work on the optimization of the Maillard reaction, and so we need to work on which kind of sugar and how much we need to add the sugar, and not only sugar, sometimes we need to And another thing which is we need to work on it and decrease it, I'm sure, because as she or he mentioned, it's not very good for health, but we are trying to keep it in the minimum. But definitely if you want to have the Maillard reaction and work it properly, you need to add the fats and sugar and protein. But the concentration is critical. So we are working on to the minimum rate to have that doesn't have any side effects or yeah. But she's absolutely right. So we are using sugar, but we need to add it. Otherwise, it doesn't work. And you cannot, after the heating, you cannot have that color or aroma or taste. That's why definitely we need to add, but the concentration is very important and tricky. Yeah.
Okay. Thank you very much. Okay,
No
last
worries.
question. I think this is a good one to wrap it up as well. And it has 3 upvotes, so people do really want to know. Given the impact of animal agriculture on the environment, would these alt meat dairy products would be more sustainable as they scale in size? How do you take it from a scientific lens or another lens? I'll pass it to Nick.
Sure, absolutely. I think Sam was covering some of the ideas about how many calories go into meat and how many, for example, beef is terribly inefficient if you just want to look at it in those terms. So I think Sam could, he's got the figures on that. Would you like to take that, Sam?
Yeah.
Yeah, if I take that from, would it be more sustainable as it scales? I think what scale will lead to ideally is commercial viability. And that's, I think, like a really, really important factor, that we need to make these products commercially viable and we need to make them taste so that people desire them. Ultimately, I think the sustainability of this sector is going to come down to the decisions that companies make. And I think it is possible to make decisions around, for example, how you use water, which is, there is going to be a significant amount of water required, how you access energy. Do you go through renewable sources or other, because there's going to be a significant amount of energy required. And I think to a certain extent, there is a necessary scale from the industry's perspective, or the industry needs to get to a point where it is commercially viable for some of those decisions to be possible, because some of them are going to be quite cost intensive, particularly from a CapEx perspective. But yeah, so I think the scale is an enabler, but for me, what's going to be the most critical for how sustainable this sector will be is the genuine intent behind the companies who are driving it. And whether, and this is going to come down to to a certain extent, a discussion around profits, as it has done around many other industries, which is, are we willing to make decisions to use renewable energy only? Are we going to make decisions to recycle water wherever we possibly can, knowing that they will incur a cost, particularly upfront, but they may be more cost effective over the long term? So yeah, I think that the scalability is an important factor, but ultimately I think it comes down to the decisions that people make. And I think the beautiful thing about this sector is that we have myriad decisions that we can make that could lead towards an inherently sustainable food supply chain in the future, which is, yeah, for me, incredibly exciting.
Absolutely. And one of the choices may be to join this, movement where we build new food systems with alternative proteins, especially in cultivated meat. So yeah, I think I'm going to wrap it up now, as I think we're past the time. But Sam, you did mention that there is a talent shortfall at the moment in this sector. And so definitely check out Cellular Agriculture Australia's pathways to, actually, do you want to share screen, Marcella? Oh, go back. Yes, Cellular Agriculture Australia's pathways tool, absolutely amazing. I wanted to shout it out one more time. Check it out if you're interested in a career in this field. In the next slide. Also, if you want to learn more about, cellular agriculture, cultivated meat, definitely go check that resource out as well. I'm going to wrap it up. But if you have any other closing remarks, feel free to speak up now as well for the panelists. If not, then that would probably be the end for session 1. Thank you so much
very
for the
much.
speakers.
Thank you very much.
Thank you very much for organizing that. Thanks.
Yeah, well done, team. You've done a great
Yes,
job.
thank you.
Thanks for
Thank you very
inviting me.
much.
Yeah.
Thank you
Great.
so much,
Thank you.
everyone.
Thank you.